Good morning, good afternoon, and good evening, everyone, and welcome to the 13th and penultimate webinar in the Neurophysiology 2023 series. Today's webinar is titled Organoid Meets Microelectrode Array, or MEA, Accelerating Drug Discovery and Development, featuring Dr. Sven Schönecker from Multi-Channel Systems and Sara Mirsadeghi from the University of Texas at San Antonio. Today, they will present their research on 3D mesh microelectrode arrays, including new developments and potential applications. Before we get started, we would like to acknowledge our partners for this series: the American Physiological Society, the Society for Neuroscience, the Federation of European Neuroscience Societies, the American Autonomic Society, and Scientist.com. We'd also like to say a big thank you to the sponsors of today's event, Harvard Bioscience and Multi-Channel Systems. And with that, I'm now very pleased to welcome our first speaker, Sven, to join me. Sven, please take it away whenever you're ready.
Hello, everybody. Andrew, thank you very much for the nice introduction. And I'm excited to talk about Organoid meets MEAs today on that nice platform. And I will give the talk today together with Sara Mirsadeghi from the University of Texas at San Antonio. And I will make the first part of the presentation and will hand over later on to Sara. And Sara will present some nice data with our equipment and some organoids. Before I start, a short introduction about myself. So I'm born in Nürtingen, Germany. It's this city here on the Neckar River. I studied biology at the University of Tübingen and did my PhD later on in pharmacology at the University of Tübingen in a cooperation with the Natural and Medical Science Institution, NMI. And I'm part of the Harvard Bioscience family since 2015. I worked for two and a half years in sales, moved on to the application team, and took over the lead of the application team later on. Since last year, I'm a product line manager from our MEA electrophysiology. And before I start, the most important in my life is my family. So I'm married to Anna. I have two sons, Bruno and Leo. Let's get started. Harvard Bioscience is a global company specialized in life science solutions, and you can basically see all of our 11 brands here. And today I will focus on multi-channel systems. Multi-channel systems is a company based in Germany in the Stuttgart area. It was founded in the 90s and is a division of Harvard Bioscience since 2014. We are specialized in electrophysiology solutions. So we have equipment for extracellular and intracellular recordings and stimulation devices. So basically patch clamp, oocyte electrophysiology, and in vitro and in vivo MEA systems. We are around 70 people. We have a big R&D department, and yeah, we are in a nice area here in Southwest Germany. A few words about the today's global footprint from Harvard Bioscience. So we have 3 core operating sites. The headquarters is in Boston, and they are focused on cellular and molecular solutions. We have a site in Minneapolis, the preclinical systems, also known as DSI. And our Stuttgart site, as I said, is specialized in electrophysiology. We have other sales offices and small centers of excellence in Europe. So Biochrome in the UK, PenLab in Barcelona, Spain, CMA in Stockholm, Sweden, and another company in Germany, Hugo Sachs Electronics in Freiburg, Germany, and a big sales office in Shanghai, China. Here on that slide, you see our MEA portfolio. It's a modular portfolio. So in the center, you see the interface board, which can basically connect to all head stages here. And once you have a system from us, you can easily upgrade to another system. Here on the left side, you see our in vivo solutions. The MEA 2100 system is a tethered in vivo system with a specialized preamplifier where you can freely select your simulation electrodes, your reference electrode, and so on, and has very good amplifiers. So it's a very low noise amplifiers and very good signal to noise ratio. Further down here, you see our wireless system. With this system, you can record from freely moving animals, and with one system, you can record from 8 animals simultaneously. Our in vitro systems, would like to start here on the bottom part, the upper part, is our single well MEA systems. For example, our MEA mini system. It has a small footprint and can basically connect it to all sorts of microscopes, so you can combine also other technologies like imaging and so on. You can place the system inside an incubator, and you can choose a different amount of recording electrodes. If you are interested in more higher throughput recordings, you can go with our multi-well MEA system. Here you have 24 or 96 well plates, and that's ideal for safety pharmacology recordings or drug screening. The system can also be combined with our MEA Express, which is an automatic pipette robot, which makes it easier to apply compounds or drugs of your interest. If you're interested in higher density recordings, we have also a CMOS system in our portfolio. Here you have 4,225 recording sensors on a square millimeter, so really high density. And we have also systems in our portfolio where you can record from pancreatic islets, and you can do some diabetes research. The reason for the webinar today is that more and more people are reaching out to us, and they are asking, okay, I want to record from 3D cell clusters, from organoids, spheroids, and I will talk about that later on in more detail. Let's talk a little bit about electrophysiology. So I would like to compare the classical methods like patch clamp and intracellular electrodes with the MEA technology. So patch clamp and intracellular electrodes, they have some disadvantages. So you need, first of all, high technical skills. You need to penetrate the cell membrane. And exactly here, MEA recordings have advantages. So you have a planar microelectrode array, an electrode on a substrate, for example, glass, and you can do extracellular field potentials. And the advantages are that you can non-invasively record from multiple cells or locations. You can do long-term recordings. It provides you with a simpler experimental handling, and you have high spatial resolution and network level insights. And at the end, if necessary, you will get a higher throughput. Here on that slide, I want to explain a little bit how an MEA chip looks like. So here you see a standard single-wall MEA chip based on a glass substrate. On top, you have a culture or recording chamber, and in the middle, you have the electrodes. The electrodes, basically, you can choose what kind of electrode you want to use for your experiments. We have mostly titanium nitride, gold electrodes, or gold electrodes coated with carbon nanotubes to keep the impedances low and to provide you with a good signal-to-noise ratio. The chips are usually isolated with silicon nitride on top, and you can choose different electrode sizes from 10, 30, up to 100 micrometers, depending on your needs. There are also variable inter-electrode distances, and at the end here on the lower left side, if I talk about the standard MEA systems, you can choose 60, 120, or 256 electrodes. The main advantage of the standard MEAs are that the chips are fully transparent, and you can combine electrophysiology with other techniques like, for example, imaging. If you need higher throughput recordings, you can also choose the CMOS chip, where you have more than 4,000 recording sensors and more than 1,000 stimulation sites. Again, there's a wide range of available electrodes and layouts, from single well to high throughput recordings. And here are some examples. So this is a 9-well MEA. Here on the right side, you see a 24-well plate. On the lower left side, you can see 3D MEA or spike electrodes, basically developed for brain slice recordings. And for all MEA chips, stimulation is integrated. And no matter if you are interested in electrical stimulation via current or voltage, or light stimulation, both is possible. What are the core MEA applications? Of course, there are more of them. This is just an example, a little bit more into the neuroscience direction. So neural network studies, synaptic connectivity, neuropharmacology, any sort of disease which is correlated to the CNS, so epilepsy research, for example, drug discovery and development, in vitro disease models, neurotoxicology, brain-computer interface studies, and of course, stem cell and organoid research. And yeah, it's time to talk about organoids, so a new dimension. Organoids are small-scale simplified organs grown in vitro, derived from pluripotent stem cells or tissue-specific stem cells. And the main advantages are that they are self-organizing 3D culture systems. They recapitulate key features of real organs, and they are mimicking the complexity of tissue and organs. And you can obtain personalized and disease-specific studies. Screening and evaluating drug responses is also possible. And of course, they play an important role in the 3 Rs, because everybody wants to at least minimize the use of animals. Why organoids? So this is a nice review from Kim et al. And you can directly see that they compared 2D cell cultures with organoids, and you can see, okay, there are definitely advantages. And it is expected that in the near future, the variety of organoid models available, coupled with advances in recording technology, so that would be our responsibility, will provide a series of powerful and efficient platforms for studying human development, physiology, and disease. So far, so good, but what is the state of the art? So, as I said, many people are reaching out to us and they want to record from cellular 3D structures. And what they did is they just used 2D electrodes. And yes, it's possible. So this is a quite exotic example. So we are looking at pancreatic islets here, so intact islets. And you can basically cultivate them on a matricel-coded MEA. And then you can record the electrical activity. You can see here. So you see nice oscillatory activity. It's a response to glucose, and that's correlated with insulin secretion. So also different glucose concentrations increase electrical activity. And you can use an MEA mini system from us, either with 60 or 120 electrodes. The problem is that you only record from the surface of the organoid or organ, and over the time there's a morphological change of the structure. So if you look on day one, they look still very round and nice, but after 2 or 3 or 4 days in culture, they are flattened and they look like a pancake. And this has, of course, a strong effect on the physiology of the cells. Another example would be recordings on a CMOS chip. Also, that is possible. And you have for high resolution, so you can even follow the propagation of the signals and so on. But you have the same problem that morphologically the cell cluster will change over the time. So it's not ideal for long-term recordings. Another option is to use in vivo probes. So you can use an in vivo MEA system from us, the ME 2100 system, where you have the in vivo probe connected to the preamplifier here. And then with the help of a micro manipulator, you can just penetrate all sorts of 3D structures. But of course, that's an penetration, so that affects the cell survival, of course, and it's also not a solution for long-term recordings. Another example would be our 3D MEAs or spike MEAs. They were initially developed for brain slice recordings, and after the slicing procedure, you always have dead cell layers. And the idea was that the spike electrodes overcome the first 2-3 dead cell layers, and at the end it provides you with better signals. And our customers, they try to use that with organoids, and it works. So you can record data, but at the end, for long-term recordings, still not ideal. And now I'm really happy to present you our new development, our so-called Mesh MEA, where we have 60 electrodes on a flexible MEA. And if you look at the photos here on the right side, you see that you have very thin filaments and a mesh structure, and then the electrodes on the junctions here. And between the filaments and the electrodes, there's a lot of free space, and you can simply grow your organoid, your cells, above on the mesh, and over the time the cells will simply incorporate the MEA into their structure, and that makes recordings from the inner parts of an organoid on several locations possible for the first time. The good news is that this MEA chip can be adapted to our existing MEA systems, and you can just place that, for example, in our MEA mini amplifier. On the next slide, you see a nice video of the mesh MEA. If you look in the center, you can see the mesh. It's difficult to see the electrodes. They're pretty small, only 30 micrometers in diameter. Then on top, the recording chamber, you can add a perfusion. You can also add a cover to keep the organoid culture sterile. And on the next slide, I can show you some nice microscopic photos. Here, the focus of the microscope was on the mesh, and on the other photos, the focus is further down, further away from the mesh, and you can really nicely see how the cells incorporate the mesh into their structure. And at the end, last but not least, of course, you also need a data acquisition and analysis tool to record from the chip. And for that, we have our multi-channel suite, a tool for data acquisition and online and offline analysis. It's pretty straightforward and simple to use. There's a simple drag and drop interface, and you can detect spikes and bursts online and offline. There's an integrated stimulator instrument, which you can program, and you can use the internal stimulation units, and you can obtain real-time signal detection and feedback. That's already everything I have to say today. I want to say thank you for your attention, and I'm of course happy to answer your questions later on. But now I'm excited to introduce you to Sara, and Sara will show you some real data with our hardware and the Mesh MEA. Thank you very much.
Thank you, Sven, for the nice introduction, and thank you all for listening. So as he mentioned, I'm a researcher and I'm a neuroscientist. So I studied developmental biology for my master's, and then I learned electrophysiology. So no wonder I'm interested in brain on chip, and by chip I mean multi-electrode arrays. And today I'm going to talk about my own journey as a researcher using multi-electrode arrays and employing different type of multi-electrode arrays in my research. And also, I want to show you some cool results about the Mesh MEA. So I'm a PhD student at Dr. Jenny Shea's lab in University of San Antonio, University of Texas and at San Antonio. But I started working with multi-electrode arrays, standard multi-electrode arrays, in my previous lab. I'm interested, as I told you, in the neurodevelopment and the electrophysiological aspects of the neurodevelopment. So first, I started growing primary culture neurons isolated from rat and mice brain. And my questions are how network connectivity can change during the development, and how we can manipulate the network connectivity, or how we can manipulate the neuronal plasticity on a chip. And I use 30 micrometers and also 10 micrometers chip in my experiments. Using mouse and rat-derived cells, we were able to keep the cells in 2D culture for more than a month or 18 days. And I did recording like every 2 days, every 4 days, and then we were able to monitor the evolution of the neuronal connectivity and activity. As you see here, at the beginning, the neuronal connectivity is not that strong, but then it starts to change the center of connectivity on the chip, and then eventually in about 20 days in vitro, the connectivity is stronger than any day. So now I'm interested in human-derived brain organoids. And as I think you all know, there are critical differences between a rodent brain and also a human brain. And to name a few, the cell type, the number of the cells, and you see we have this zone of progenitor cells, which make more cells and which give rise to this gyrus in the brain. So human brain is definitely different from a rodent brain. And we're interested to generate this humanized brain using iPSCs, as Sven mentioned. These are pluripotent stem cells that we can actually differentiate these pluripotent stem cells to every neuronal cell types in the brain. And we can also use the pluripotent stem cells from healthy donors and also people with different genetical background or congenital epilepsy or Alzheimer's disease. So as a researcher, depending on my question, I go for patch-con recording or multi-electrode array recording. Here, as you see here, since I'm interested in neurodevelopment and I'm interested in evolution of the network and neuronal electrophysiology over time, if I want to address the membrane properties of a neuron, within the development, I choose patch-con recording, because patch-con recording provide me everything about the voltage-gated ion channels, everything about the network between only two neurons. But if I want to ask any question about the synaptic plasticity, about the different types of neuron contribution in the network and the development of this network, I would prefer to go with multi-electrode array. And today, I'm going to talk about my experiments using multi-electrode arrays. So yes, again, the question is, just imagine that if I grow brain organoids, humanized brain organoids, can I have this plot and can I compare the evolution network connectivity in different days of the human organoid? With this question in mind, I started using the standard 2D organoids and also 3D spike organoids. So I put, as you see here, this spherical shape of the organoid. I located the organoid on these 2D electrodes and also the 3D electrodes. And I was able to do some accurate recording for several hours. But the question still remains. I want to mention that my question, at least, is still remain, because I'm interested to be able to record from one organoid over a long-term period. These organoids, some of these organoids, we keep them for more than a year. So just imagine if I wanna ask a question about the neural network development within that year. It's not possible to do that using these two type of chips. So I can just have electrode organoids, locate different organoids in different time points of development on these electrodes. But I'm not able to record from one single organoid over time. So we need another solution, and the other solution for me, at least, was this Mesh MEA. I'm going to elaborate on each experiment. This is my criteria of recording. This is the stage criteria of recording. Of course, we save the raw data with all the frequencies and action potentials, but on a stage, I'm interested in high pass 400 Hertz spikes, which is reflective of action potentials outside of the cell. And also, I'm interested in local field potential, the frequency between 5 to 10, but the software allows us to filter the spikes and filter the data on a stage with different frequencies that we are interested in. So in the first experiment, as I told you, I put the organoid on the standard 2D electrodes. On top of these standard 2D electrodes, I usually do recording inside the incubator for 30 minutes at least. And here you can see that I wasn't able to add more media, more than 200 microliters, because I want to keep the organoid stable on top of the electrodes. And of course, I was able to record from the surface of the organoid, and we have the raster plots. I mean, the spike times here. As you see, this plot shows the number of spikes in y-axis, and also this is 5 minutes of that 30 minutes recording in x-axis, and each tiny dot here is representative of one event, one spiking event. So after 30 minutes of recording, the organoid changes morphology, and it becomes like a pancake. And then after that, I have to throw away the organoid. So what is the advantage of using these 2D chips recording from brain organoid? Of course, I can record from the surface, and I can do some accurate recordings for 30 minutes or several hours using organoids with different time point of developmental neurodevelopment. And of course, it's accurate recording. But the remaining challenge for me, at least, is if I can record from one organoid multiple times, and if I can implement perfusion system, or I can record from organoid for more than a day or for more than several hours. In my next experiment, I use the 3D spike electrodes. I put the organoid on top of these 3D spike electrodes. Since these electrodes can penetrate into the organoid, I was able to add more media to the culture. But again, and you see here, I was able to pick up the spike times. I have the raster plots here, and I was able to record for several hours. But again, I was able to partially keep the brain organoid anatomy. So 3D spike electrodes provide me a platform to be able to penetrate to the organoids and be able to record from internal regions. In addition to surface, it gives us the platform to be able to record from internal regions of the brain organoid. And of course, I could add more media, and I think we can employ perfusion system for longer period of recording, because the organoid is pretty stable on these 3D electrodes. But still, my question remains how I can record from one single organoid over time. So since I was the beta tester, one of the beta tester for the mesh electrodes, I first recorded from the mesh electrodes without any organoids. So this is the view of electrode signals, electrode noise level, because I wanted to make sure that if the electrode noise levels are normal and I'm not recording after locating the organoid, I'm not recording any artifact. And as you see here, the electrodes were cleaned. And after I added the organoid, I located the organoid on the Mesh MEA. Right after that I did that, I was able to see some spiking activity and action potential generation on the Mesh MEA. And then I kept the organoid on top of this Mesh MEA. For this preliminary experiment, I used the same organoid that I recorded using 3D spike electrodes on the Mesh MEA. I use the same organoid because this is my quality control, of course. So I want to make sure that the organoid is actually active. So after 2 days on Mesh MEA, as you can see, the organoid stayed on the Mesh MEA. Some of the cells tend to migrate around the mesh and around the electrodes. And I was able to pick up even more spiking activity using Mesh MEA, as you see in the raster plots. And of course, I tried again to keep the organoid on top of these mesh electrodes, and I monitor the cell migration and the health of the brain organoid on the mesh electrode. As you can see here, within 1 week, the neurons start migrating around these filaments, around the mesh. That means that the cells are freely migrating and the cells are healthy. As you can see here, they can grow axons, they can grow dendrites around the mesh electrodes within 1 week. And I hope to keep these organoids on top of these mesh electrodes even for more than 1 week, for months. So as a researcher, as you all know, in the brain organoid field, there are many questions and there are multiple challenges that we have to overcome. In previous studies in Lancaster lab, they generated these big organoids, these cortical organoids, and then the idea for them, since the organoid doesn't have blood vessels, of course, and when we grow the organoids bigger than normal, bigger than usual, we see some necrotic core usually inside the organoid. So the idea in this study was to make sections from a big organoid and put that section on a mesh structure. They did this, they make the section, they put the organoid on a mesh structure, they try to keep the media level at the bottom so that they make oxygen interface, air interface, and nutrition interface for the organoid. And surprisingly, after doing this research, they realized that they can keep the anatomy and the neurons here, as you can see here, not only the neurons grow longer axon, but also they make some axonal tracks. But they didn't have the electrodes inside this mesh, so they weren't able to monitor the electrical activity of the organoids using this air interface strategy. So hopefully in our next experiment, we'll be able to actually find something different in electrophysiological development of the organoid. So not only the mesh structure can compensate for the blood vessel loss for the brain organoid because of the hole inside of the mesh, but also it gives us the platform that we can monitor the electrophysiological development and connectivity and evolution of the connectivity of the network on the electrode. And for me as a customer, so Sven showed you the platform, but from all of these amplifiers and products in our lab, I only have this MEA 2100 mini, and I use all this variety of multi-electrode arrays, recording using the same platform, using the same head stage, and for me. It's the plus as a researcher that I can use the same platform, but I can just buy variety of the multi-electrode arrays for my research. You may wonder what is so we just imagine that we grow the brain organoids separately. We have different types of multi-electrode arrays. Now we have the mesh electrodes. Hopefully I can use this mesh electrode for long-term recording in my research. But previously in our lab, we were able to make organoids from healthy donors and also patients with epilepsy, with genetical epilepsy. So in general, in Shea Lab, we are focused in epilepsy. And we are interested in modeling epilepsy, mostly epilepsy on chip. And recently, we were able to record using 3D spike electrodes, and we record from healthy patients, healthy donor brain organoids, and also patient-derived brain organoids. And I was able to pick up a higher synchronized spiking activity in the brain organoids that are derived from seizure patients. And as you know, higher synchronized spiking activity is an indication for seizure. So if you're interested to know about this topic and how we compare between control organoids and also patient-derived organoids, follow our papers, because our paper is now under review, and I think in 2 weeks we'll have the bioarchive release for this paper. I would like to take a moment to appreciate my colleagues here in Shea Lab. All of these people are experts in brain organoid development. These people can make the most functional brain organoids, which I like as an electrophysiologist. And I would like to appreciate my former PIs, Sahar, Michele, and my friend Ali, who helped me with the analysis. And of course, I would like to appreciate my current PI, Dr. Jenny Shea, who supported me for my ideas and led me to participate in this webinar. Thank you for your attention, and I'm happy to take the questions.
So without further delay, let's welcome back Sven and Sara onto webcam. All right, welcome back both. And jump right in. Are we ready for some questions?
Yeah, sure.
Perfect. A great one here. I think it's probably a common question. How do you actually grow the organoids on top of the mesh MEA? And are you using any specific coating materials, laminins, matrigels, etc.?
Yeah, I can take that question. So the way that I usually grow the organoids on top of the mesh, yes, I use matrigel coating as normal, using normal protocol that we have. And then after that, I locate the organoid on top of the mesh, try to keep the media beneath the mesh for about an hour in the incubator. And after that, the organoid is totally attached to the mesh, so you can add more media.
So within an hour, it's attached?
Yeah, since I'm using matrigel, I didn't have any problem after 1 hour.
Okay, wonderful. So once you've got, another question here is about manipulating the neurons in the network. Once you've got it established, how are you able to manipulate it? Do you use stimulation, etc.?
So the system, as Sven mentioned, you have voltage current stimulation, and also I think you have optogenetic option as well. So you can just add an accessory for LED exposure to the organoids with the cells expressing the channel rhodopsin. I usually use voltage stimulation. So the MEA 2100 is integrated with a stimulator. So, and in the software, you have option to to design a protocol for stimulation. I mean, different timing, the shape of the stimulation. And I think these are the options for stimulation and manipulation.
Okay, great. So basically, depending on your specific research question, you tailor your stimulation protocol to address that. Oh, that makes sense. One of the things you mentioned was the modularity. I think you both mentioned the modularity of the MEA systems. question here is whether it is possible to record from more than one MEA mini system at the same time.
Maybe I can take that question. I had mentioned that the portfolio from us is entirely modularity. So you can basically use the same interface board for different head stages. But what you can also do is, if you have an MEA mini system, for example, with 60 electrodes or 120 electrodes, you can add several other head stages to the same system. So we have at the end options to record from 8 systems simultaneously. So you can easily increase your throughput, and you can just add other head stages. If you have an entirely different head stage, for example, a multi-well head stage and a mini head stage, then you can only use one at a time, and then you can just unplug the one and you can use the other system.
Okay, fantastic. A great question here. I think you touched on this a bit, Sara, but can you see differences in the actual morphology of the organoid when you're using standard 2D electrodes and the Mesh MEA after longer periods of time?
For sure. Yeah, for sure. When I put the organoids on the 2D, after a while, they become a pancake. But then on 3D, it's better with the spike electrodes. You can maintain the morphology for longer period. With the mesh electrode, since this is the dynamic of the cells, they tend to migrate. Of course, the morphology would be changed, but at least I'm sure that they are healthy, the cells, because the most important thing for me is to maintain the physiology of the cells within the recording. So I used a 3D and Mesh MEA. I'm satisfied more with 3D spike electrodes and Mesh MEA for brain organoids.
So in essence, you're saying, I guess, if just to paraphrase here, because I'm certain there's a lot of interest in the long-term recordings. Obviously, it's a great appeal of the system. With the 2D, you do see the morphological changes, but it's associated with a loss of cell viability, largely. Whereas with the mesh, then you are seeing morphological changes, but it's related to the development of the organoid rather than the dying off of the cells. Is that correct?
Yes. So with mesh electrodes, I'm not doing anything special. It's the cells that moving around. So it's not as invasive as other electrodes. And depending on the question, I think all of the chips are useful employing our experiment depending on our question. But I'm more satisfied with 3D shank electrodes and mesh electrodes for brain organoids specifically.
Right. Yeah. Oh, fantastic. What's the longest that you've looked at an organoid with the mesh system?
With the mesh system, it was about 1 month.
Oh, wow. Okay. Has anyone tried something really extreme with them that you're aware of? Is there a world record holder for longevity?
Sven?
As the mesh MEAs are pretty new, the world record is Sara's recording so far.
All right. So with a bit more time, and all entries are welcome, I suppose, right? New record holders are welcome. Fantastic.
You should know that I'm tough.
Yeah. A great question here is, you touched on this a bit with the optogenetics, I think, but can you combine MEA recordings with imaging methods?
Oh yeah, since all the chips are made of glass, you can just move the chip after recording under microscopy. I did even live imaging after some recording because the organoids that we have in the lab, there are tracer expressed in the cells. So we have glutamatergic and GABAergic cells with different tracers. So after recording it, I did it. I moved the organoid on the chip under microscopy and did some live imaging. And also you can implement calcium imaging as well.
Okay, amazing.
Yeah, I did some calcium imaging too.
Yeah, I would like to add something here. So, and you can even do that during a recording. So,
Yes.
when I was back in the lab, I did electrophysiological recordings on standard MEAs with pancreatic islets, and at the same time calcium imaging. And the same is basically also possible with the Mesh MEA or with the Spike MEAs. So, because of the small footprint, you can just integrate our systems on top of any microscopes.
Okay, fantastic. Are there any specific sort of tips, recommendations, limitations at all with it, or is it just it sounds relatively straightforward?
Limitations depends also a little bit what kind of wavelengths you want to use. So I'm not an expert in calcium imaging, but it depends what light sources, what wavelengths you use. And also for that, we have an answer. So for example, you can use specialized chips with a very thin glass substrate so that you can easily come through with all of the wavelengths of your interest.
Okay, fantastic.
And one last thing,
Sure.
our interface board has also digital in and output, so you can synchronize our system with other systems, and you can communicate with them, and you can synchronize the recordings.
Okay, yeah, that's probably very important with the timescales involved. Okay, fantastic. Another question here is relating to comparison between the different systems. Are there differences between the electrodes from a Mesh EAA and the standard MEAs? And as all electrophysiologists will be interested in this, if so, is there a difference between the signal to noise ratios between the Mesh and standard chips?
That's a good question. So first of all, as I mentioned in the talk, we are using different electrode materials. Usually for neuroscience or for smaller amplitudes of the signals, I would recommend titanium nitride or p-dotted gold electrodes. Titanium nitride has the advantage that they are very durable and reusable, so they are very stable. And we use titanium nitride for the Mesh MEAs as well. And also the exact same diameter for like for the standard MEA. So it's 30 micrometers in diameter. And as Sara nicely showed when she tested the Mesh MEA in her lab, because she wanted to make sure is how is the baseline, how is the background noise, and so on. And we have exactly the same background noise like with the standard MEA. That was a very important first feedback for us. And then later on, when we saw the first results and the signal to noise ratio, it is absolutely comparable and maybe even better because the cells can grow around and have a closer contact to the electrodes. So that is really important for the Mesh MEA, that the electrical background noise is absolutely comparable to our standard MEAs.
Okay, fantastic. I mean, the signals did look very clean. The signal to noise ratio looked great in those examples Sara gave. Is there any filtration on that at all, Sara? Is that just the raw signal coming out there?
No, that was on a stage. I usually filter, as I showed you, my criteria for recording is high pass 400 and also a bandwidth of theta, because I'm interested in that frequency. And I'm interested, for me, it's important to pick up the action potential reflection outside of the cell. Because I want to compare, we mostly want to compare the control organoid activity to epileptic organoid activity. So the number of spikes and also the synchronicity of the spikes is important. What you saw in my presentation is on a stage, but I save just to know what I have in my data. So I can, because it's large amount of data. So if I see no action potential, I don't proceed with the analysis process.
Sure.
But of course, we save the raw data to be able to filter out of, like in future study, filter out different frequency.
Sure. Yeah, no, that's fantastic. Maybe too preliminary to ask this, but have you been able to detect any differences in the epileptic organoids?
Yes. As I mentioned in my last slide, we have one of our exciting publications coming out from Shea Lab. The paper is under review, and the bioarchive version would be out in 2 weeks, I think. And yes, we were able to pick up differences between control organoid and also the organoids derived from patients with genetical epilepsy, and they're totally different. Hyperactive, synchronicity. Yeah, that's
Fantastic.
exciting.
Obviously, it's not going to be ready in time. Isn't ready in time for today's talk, but we can potentially add it into the resources as well in a couple weeks' time when it does get.
Maybe the next webinar then, Sara, for you.
There we are. Yeah. Great question here. Are there any other specific challenges and opportunities when using the MEAs to record from organoids? Specifically with the added benefits of being able to look at the 3D structure and the cellular heterogeneity.
So we cannot overcome the cellular heterogeneity of the organoids because that's biology. But in terms of electrophysiological recording, I remember that I have this challenge or many people, I would say, have this challenge to keep the attachment of the organoid to the electrodes. And the 3D, that's why I'm saying that I'm satisfied with the 3D spike electrodes, because that's easier to keep the organoid on top of the electrodes and have the best attachment for recording. And the other issue would be sometimes you face media evaporation, because I record inside the incubator, but for that, I also use the specific membranes from company, a cap. So when you cap the chip within the recording, during the recording, you don't face any evaporation. I think those are the challenges that I faced.
Have you been using? You mentioned perfusion systems. I wasn't sure if are you using those in your systems as well, or any comments?
Yes, I'm about to establish an automatic perfusion system because I'm also interested in drug testing. Our lab is interested in drug testing, so we would like to test different drugs on especially epileptic organoids, because most of these organoids are refractory to any medication. So we're hoping to be able to perfuse the organoids while recording with some of the selective drugs. And as I think, Sven, you mentioned this, you can add an accessory for perfusion to the recording system, and it can be automatic with the trigger. It can be automatic with the recording.
Yeah, absolutely. So we have perfusion systems and pumps, and they can be triggered via TTL, so digital in and outputs, which you can combine with our systems. And also on the Mesh MEA, I need to be entirely honest, it's a very new product. So we started with preliminary recordings, like Sara did, up to 1 month. The next step would be to add perfusion. And also one important thing is what you can do with the Mesh MEA. You can do an air-liquid interface, because that's very important for the organoids to survive. And also that should be possible with perfusion, but we are still working on that.
Okay, yeah, watch this space. Yeah, I would imagine oxygenation and level of oxygenation would be really important variable here. On that note, the next question ties into that quite well. We're almost out of time here, but I think we can squeeze in another one here. I think we've kind of answered this, but maybe if you'd like to elaborate it on further, can MEAs be used to investigate long-term changes in electrical activity? For example, in the study of developmental processes, disease progression, etc. And I think we've talked about it in some depth already, but do you have anything else to add to that? And maybe new opportunities for studies that these enable?
Well, that's the goal. That's the plan, to achieve that, using this mesh multi-electrode array to be able to record the long-term changes within the organoids. In my case, like, as I already observed differences in network evolution in the control and epileptic organoids, so I'm waiting for the Mesh MEA to be optimized enough.
Yeah, Yeah, indeed. So it looks promising. Essentially, all of the signs point to yes, and then now it's just a matter of time to accumulate the long-term recordings.
Yeah, because with the other ones, so with the like 2D and spike ones, I have to do accurate recording. So I recorded from several hours, and then after that, you have to remove the organoid, use the organoid for other like molecular study, and that's it. But I'm interested to know how the network evolves, and our data that you'll see in our paper is promising that there is different, even from the very beginning of the development, different neurodevelopment in the brain organoid with different genetical backgrounds.
Okay, well, a great plug, a great teaser. I think we're all probably looking forward to seeing the paper now. It'll be very interesting to see how this develops moving forward, and other potential applications, other disease models. It's fascinating stuff. Unfortunately, I think we are out of time. But Sven, Sara, I'd really like to say thank you for sharing all of your insights with us today. It's been an absolute pleasure having you with us.
Thank you very much.
Thank you very much, both. Another quick thank you to our partners for helping make this series possible, and particularly today's sponsor, Harvard Bioscience and Multi-Channel Systems. And in closing, we hope you enjoyed this Inside Scientific webinar, and we'll see you again next time.